I'm Yue Huang from UBC Bioinformatics Master Program. This work is done by multiple members from PATH Lab and also Pavlidis Lab from UBC. Let's look at the introduction first. Phospho flow cytometry can measure the phosphorylation states of different proteins inside cells. And it has two crucial advantages of the flow cytometry. The first is single cell resolution. The second is multiple measurements of different proteins. May be heavily influenced by the flow cytometry. A population level data analysis method is still widely used in phosphoflow as well. That is calculating the fold change of MFI before and after a stimulation. Though this method is quite simple, it overlooks the heterogeneity of cell populations. So here, we want to develop a single cell level data analysis pipeline for phosphoflow datasets that can recognize this heterogeneity and can also extract additional information from the datasets. So let's look at the methods and results. In the first section in figure 8, as we can see, after single cell standardization, cells from different groups are plotted on the plots denoted by different colors, either P10 Y type as an experimental group or the empty control as a control group, indicated by two different colors. And the x-axis is just the transfection level, and the y-axis is protein phosphorylation level. So here is PKT, for example. And after single cell standardization and also using the Loess regression to show the general trend of the data denoted by the curves, we have two curves in each group because we have two replicates for each group. And after we plotted all the single cell and also the Loess regression, we will use two breakpoints indicated by the two vertical dashed lines to separate the whole cell, the cell populations into three subsegments. In each segment, each segment is called a linear-like segment. And for the middle part, we will call it the linear response region. So in each of these three segments, we will build a linear mixed effect model, and the result is showing as a black line. And the slope of the linear mixed effect model in the middle part, we will call that slope effect size. So in figure D, we cannot just plotting the effect sizes of different groups, not only P10 Y type, but also RAP Y type and other 5 P10 variants as well. So we can see P10 Y type and RAP Y type are showing very different effect sizes on PKT, possibly because they're acting on very different sections of the signaling pathways. And also, we can see that the 3 loss of function mutations of P10 are showing much weaker effect sizes compared to the P10 Y type towards PKT. So in figure C, we are not just showing you PKT, we are showing you the results from the other 3 phospho macroproteins that we measured in figure C as well. And in the second section, I want to show you how those dependent analysis of signaling macroproteins can reveal code deactivation and also better distinguish variant functions. So to get the plots in figure B and figure C, we can look at figure A first. It's showing you how I got these plots. So in the middle parts, in the middle segment, we will draw 18 even dashed lines, vertical dashed lines. Each of these dashed lines will have 3 intersection points. The first intersection point is with PKT Loess regression curve. The second is P4E PP1 Loess regression curve. And the last intersection point with x-axis, which is the transfection level. After we got these 18 points, each point will have 3 attributes. To plot these 3 attributes, we can plot the 2 marker proteins as 2 axes, and we can use the point size to reflect the transfection level as shown on the plot. So we can plot the 3 attributes on a 2D plot. Not just P10 Y type, we can also plot this kind of, we can also draw this kind of plots for other P10 variants as well, showing in figure B and figure C. So in figure B, we can see that these 3 guys shows code deactivation towards this macroprotein pairs. These 3 guys are P1048, the gain of function mutation of P10 Y type, and also the Y type like mutation, the Y type like mutation of P10, which is P10 D268E. D268E. And in figure C, we kind of show you how it can better distinguish the variant function because it separates the loss of protein phosphatase function variant of P10, which is P10 Y 138L. And the other 2 loss of lipid phosphatase function variant of P10 are clustering close to the main groups, while the PTEN Y138L stands alone, as showing as the green points here. And in the last section, I want to show you how single-cell correlation analysis of signaling macroproteins can identify divergent signaling driven by different SG genes. So in Figure A and Figure B, we both plotted PTEN wild-type transfection and also RAB wild-type transfection and MP control. But in Figure A, we can see that these three groups didn't show much difference for these macroprotein pairs. But in Figure B, PTEN wild-type is showing you a difference. To represent this difference, we use we first calculate the coefficients of different macroprotein pairs for all three groups, and then we will use Fisher R to Z transformation to get you a Z-score. That is just comparing PTEN wild-type with MP control and also RAB wild-type with MP control, just to show how different the coefficients between PTEN wild-type and MP control are. So if you are seeing a right Z-score, it's showing you PTEN wild-type transfection increased the coefficients, correlation coefficients of these two macroproteins compared to the MP control. While the blue color just indicates RAB wild-type, the transfection of RAB wild-type just decreased the correlation of these two macroprotein pairs compared to the MP control. And we can see that PTEN wild-type can act quite differently compared to the RAB wild-type on changing the protein macroprotein correlation. Okay, I think that's all for the method and results. So going back to the conclusion, our results demonstrate those response and molecular pathway interactions are available from the previous classical data analysis method of phospho flow. And also our approach manifests strong promise in varied function measurements and also molecular pathway inference.